am Nina Milan, and I am here with Adele Myers, who will share fascinating research on women health brain. But before I hand it over to Adele, let me give you some context, and more precisely, some history around this lecture series. Because even though this is the first talk in Santa Barbara, this lecture series is actually much older, and it started 50 years ago, more than 50 years ago, just after the Summer of Love in San Francisco, Haight-Ashbury. Because during the Summer of Love, there was a woman called Sammy Sunchild who had just acquired a historical hotel called the Red Victorian. And Sammy Sunchild had a PhD in societal changes, and she dedicated herself to make the Red Victorian an international peaceful hub, where people from all over the world would come and gather to discuss interesting ideas around breakfast conversations. And now these breakfast conversations actually grew much, much larger, such that every week 20 to 30 people, neighbors from Haight-Ashbury, would come to the Red Victorian and discuss anything from environmental justice to actually fundamental science like quantum physics. And if you want to know more about this topic, there is actually a very serious book called How the Hippies Saved Physics, which I recommend. Anyway, unfortunately in 2013, this woman, Sammy Sunchild, died, and she donated the Red Victorian from Haight-Ashbury to a non-profit with only one wish, which is that whoever was going to own or rent that place should continue her legacy. And so a new group of people took over that place, and I was lucky to be one of them. We volunteer our time to have free lectures every week for the neighbors of Haight-Ashbury while managing that hotel. Now, unfortunately, again, because of COVID, the Red Victorian stopped being sustainable, and we had to close it so that its residents left it, and actually most of us left San Francisco. I came here and started a new job as an assistant professor and research director at UCSB, for example. But the fact that we're not in San Francisco anymore doesn't prevent us from continuing the legacy of Sammy Sunchild. And that's why today I'm very honored to be reviving the Curico lecture series, Curico for Curious Conversation. I want to thank Brass Bear Brewing for allowing us to have this event, and above all Adele Myers for being the first speaker of this new revived lecture series. And let me tell you how lucky we are to have Adele Myers here. Adele Myers is a scientific superstar. She's a second-year PhD student in physics in our lab at UCSB. And she's developing a fundamentally new theory of AI to extract scientific knowledge from biological shapes, including brain anatomies. Now, she's published internationally. Her work is world-class. She's received several awards, like the Barry Goldwater Scholarship, and 2 fellowships from the National Science Foundation, including the so-called GRFP, which is arguably the most prestigious one in our country. So thank you so much, Adele, for agreeing to speak today, and the floor is yours.
A very, very generous introduction. Thank you, Nina. And again, thank you, Brass Bear Brewing, for letting us host our talk here. And most of all, thank you to all of you for taking your Tuesday, or accidentally taking your Tuesday. I'm talking to the book club group over there, who are here to listen to me talk about the female brain. It's really, really awesome to see you all here. So as Nina was saying, my name is Adele Myers. I'm a second-year PhD student in the physics department at UCSB, and I am doing my PhD in the BioShape lab at UC Santa Barbara. I'm also a member of the Women's Brain Health Initiative, and because the Women's Brain Health Initiative was inspired by some of the recent studies about Alzheimer's, that is where we're going to start our talk today. This is a brain with Alzheimer's. Right now, you're looking at a time-lapsed video of the disease progressing in a human brain. One thing I want you to notice is that the brain is decaying. It's shrinking its volume. And if you know anything about normal biological aging in humans, you will know that this, in some senses, is normal. When a human brain ages, it does shrink. However, in Alzheimer's brains, this shrinkage happens much more rapidly. So in Alzheimer's, the visual aspect is that it shrinks, but this also is associated with memory loss, inability to communicate, change of personality, and death. And unfortunately, this disease is not going away anytime soon. For example, between 2000 and 2017, deaths from heart disease have decreased 9%. However, deaths from Alzheimer's have increased 145%. So this is a very serious problem that we should be thinking about. And there are three things that we know about Alzheimer's. One is that there's no cure. You may have heard about several drugs that have come out for Alzheimer's, but these drugs do not cure Alzheimer's. They merely treat the symptoms. The second is that it's linked to specific genes. And the third is that it's more common in women than it is in men. According to the Alzheimer's Association, almost 2/3 of Americans with Alzheimer's are women. Of the more than 6 million people aged 65 and older with Alzheimer's in the US, nearly 4 million are women. At first, scientists attributed this to the fact that women live longer than men, since age is the greatest known risk factor. However, now we're seeing that this doesn't explain all of the data that we're seeing. So, what could be causing women to develop Alzheimer's at a higher rate than men? Well, we know that there are some biological differences. We know that the rise and fall of female sex hormones cause major biological changes throughout the female lifespan. For example, the rise of ovarian hormones at around the age of 12 causes female puberty. And then small variations in the ovarian hormones during the reproductive years causes the menstrual cycle, and the decline of female hormones at around the age of 50 causes menopause. So, what are these hormones that are at play that are causing these changes? The main players are estrogen and progesterone. And you may know these because they're famous for driving the female menstrual cycle. For example, estrogen peaks at around the time of ovulation, and menstruation happens after the progesterone peak. They also play a role in pregnancy, as they both increase during the course of female pregnancy. So, we see that estrogen and progesterone play a major role in nearly every single biological event in a female's life. We see that it plays a role in puberty. We see that it plays a role in the menstrual cycle, pregnancy, and menopause. But we don't really understand what effect these hormones have on the female brain. For example, we know that these cause the menstrual cycle, but we don't know why these variations in hormones cause PMS in some women. PMS, for anyone who doesn't know, is premenstrual syndrome. And in some women, it causes severe depression for several days out of the month. And we don't know why this happens in some women and not in others. We also know that they play a role in pregnancy, but we don't know how to predict whether a woman will experience postpartum depression or not. We also know that the decrease in female sex hormones causes menopause, but we don't know how to explain that 60% of women who've experienced menopause also experience cognitive changes. And we don't know why before menopause, women are less likely than men to have Alzheimer's, and afterwards, women are more likely. So, what could be causing women to develop Alzheimer's at a higher rate than men? This is a difficult question to answer because there's a lot that we don't know about the female brain. So, now, why is it difficult to answer questions about women's health? The answer is pretty simple, and it's lack of research. Until the 1990s, in fact, only male rat brains were studied in animal clinical trials because they thought that the variability of the female menstrual cycle would skew their findings. However, this practice was not even founded in fact, because the male brain varies the same amount as the female brain. While the female brain varies over the course of 1 month because of her menstrual cycle, the male brain varies over the course of 1 day because of changes in testosterone. Testosterone is higher in the morning and lower in the evening. So, even though women's brains vary just as much as men's brains, this misconception caused a giant gap in female brain research, and this had consequences. The consequences are that men's brain research is used to diagnose women. And 80% of drugs and medical devices that are pulled from the market by the FDA are done so because of adverse effects in women. Because women are being given drugs that aren't designed for them in doses that are not designed for them. And unfortunately, this trend has continued, because even after the 1990s, from 1995 to 2020, studies on women's brain health factors constituted only 0.3% of brain imaging literature. If you want to know what 0.3% looks like, for context, it's the tiny little black bars at the very bottom of this graph. It's so small that it looks like the x-axis. I'm right next to the screen, and I can barely see the bars. That's what 0.3% looks like. So even though 50% of the world's population experiences menstrual cycles, pregnancy, and menopause, only 0.3% of the literature researches these things. How can we possibly understand women's brain health without studying women's brains? So again, I ask, what could be causing women to develop Alzheimer's at a higher rate than men? This is certainly not a thing that we're going to be answering in this presentation, or possibly in the next several years, because first we have to make up for a lot of lost time. First, we need to know how the menstrual cycle, pregnancy, and menopause affects the female brain, which essentially amounts to understanding how these sex hormones are affecting our brains. And for that, we need data. The first female to ever measure the effect of hormonal changes on her brain over 2 menstrual cycles published her results in 2020. This woman was Laura Pritchett. She was a UCSB graduate student in Emily Jacobs' lab. And every single day for a month, she took her blood, measured the hormones in her blood, and took brain scans. She took an fMRI scan and an MRI scan. And she found that brain function was heavily tied to sex hormone fluctuations. But this study only took data from one woman. To truly understand trends in female brain health, we need more female brain data. And this is why Emily Jacobs, a professor at UCSB, has founded the Women's Brain Health Initiative. The Women's Brain Health Initiative is the very first UC-wide, plus Stanford, initiative to tackle this issue. The goal is to try to scan as many brains as possible across the state of California and hopefully understand the female brain a little bit better. And they've been very, very successful. So far, the initiative has only just begun, and they've already scanned hundreds of pregnant female brains. This is a huge accomplishment, a huge step towards understanding the female brain during pregnancy. But now we're running into a new question, and that is, what is the best way to analyze all of these brains? Because we need a method that can analyze temporal data, which is data over time, and we also need a method that can analyze complex shapes. As you can see, the brain is a complex shape. So that is why Nina and I are here in the BioShape lab. We develop mathematics and software to analyze complex biological shapes, such as cell outlines and brain surfaces. We recognize that human organs and cells and brains, their function and their health is often tied to their shape. For example, in a cell, the shape of a healthy cell, the outline of a healthy cell, is relatively smooth, whereas the outline of a cancerous cell is spiky. Similarly, in the brain, you saw with your own eyes that the shape of an Alzheimer's brain is changing much differently than the shape of a brain that is healthy. So, by looking at biological shapes, we can determine things about them like their health, whether they're healthy or not. So that is why Nina and I have teamed up with the Jacobs Lab at UCSB to try to come up with new biological insights for women's brain health. And our first project looks at the hippocampus. Why do we study the hippocampus? Well, first of all, the hippocampus is a substructure of your brain. Let's just start with that. Why do we study the hippocampus? The hippocampus is the area of the brain most sensitive to sex hormones. It's also the area of the brain that shows the earliest signs of change in the earliest stages of Alzheimer's. We'll build off of work done by the Jacobs Lab. First, we'll build off of Laura's work, which you saw before, and we'll also build off of Caitlin Taylor's work. Caitlin extracted Laura's hippocampus from every single day, and she measured the volume of each one of the substructures. And what she found was that specific substructures increased their volume as progesterone increased during the menstrual cycle. So, we know that the substructures of the hippocampus change volume in response to progesterone. But can we show that the hippocampus changes its shape in response to progesterone? This question has never been answered before. So, first, Nina and I modeled the hippocampus. Well, we extracted the hippocampus and created this animation over the menstrual cycle. Then, and you can kind of see that it's changing. So, yeah, there might be something there. Then, we showed the hippocampus in response to progesterone. And as you can see, there is, in fact, a shape change. And there is a characteristic shape change. So you can see that as progesterone increases, the shape of the hippocampus shears. So even though with volume studies, they were unable to show that the entire hippocampus changed its volume, they were only able to show that substructures changed their volume. Now, by introducing shape into the equation, we're able to analyze the entire hippocampus and show that it changes its shape. Why is this exciting? Well, first of all, this is the first time that research has shown that the shape of the hippocampus changes in response to progesterone. And also, like I said, previous volume-based research was unable to prove that the whole hippocampus changes in response to progesterone. So we're introducing a new tool because the old tools were unable to show something was changing with the whole hippocampus. So, why has no research focused on the hippocampal shape before? Well, it turns out that changes in biological shapes are quite difficult to quantify. So usually people use alternatives like volume or convexity or circularity, but these are pretty coarse measurements and often are subject to human error. So they haven't been using shape, but shape encodes valuable information about the structure of the whatever we're studying, and disregarding the shape throws away this information. So if we're only thinking about the volume or the circularity or something, we're throwing away a lot of information. So how can we quantify shapes in biological objects, and specifically changes in shapes? This is actually a pretty difficult question, because we know how to quantify differences between some standard shapes. For example, the circle. We can describe everything about the circle with just the radius. If you give me a radius, I will be able to give you every bit of information about a circle. So therefore, we can compare two circles based off of their radii. Similarly, for a rectangle or a parallelogram, if you give me the base and the height, I will be able to tell you something about the parallelogram. Or with the cylinder, if you give me the radius and the height, I will be able to tell you everything about that cylinder, and I will be able to compare two cylinders with those two bits of information. But how in the world are you supposed to quantify differences between two hippocampi? These are extremely complex objects. We can't just say the radius. We can't just say the height. Like, they're pretty complex objects, so how are we supposed to describe them? This is exactly what my research focuses on. My research extracts biological insights by analyzing biological shapes. And it does so in 4 relatively simple steps. First, we take a set of shapes changing over time. So, for example, a brain, or a hippocampus, or a cell. Then, in the case of a 3D shape, like a brain, we sample the surface of the brain. And all of those sampling points together create a mesh, which you can see in this green mesh-looking thing. Okay, this is where things get a little bit more complicated. So we have a bunch of shapes evolving over time, and with each shape, we have a mesh that coarsely describes the surface. So how do we quantify differences between two meshes? For this, I'm going to introduce something a little bit different. And it is a shape space. In the shape space, every single point in this theoretical, mathematical space is a different mesh. So for example, you see here that one mesh is represented by one point, and then a mesh that's slightly different is represented by a different point, which is at a different location in this space of brains. So a mesh that is very different from the left shape would be very far away in this space of brains. But a mesh that's maybe only tweaked a little bit from the mesh on the left would be pretty close. So because of this, we can quantify differences in biological shapes by measuring their distance from each other in this shape of brains. This is an extremely powerful tool, and it's very, very exciting work. One note that makes it a little bit more difficult is that this space of brains is a curved space. So traditional machine learning methods don't really work in this space. And that's what Nina and I are working on. We are using differential geometry to develop methods to analyze data in this curved space. So, how can we characterize a shape that's evolving in this space? Well, we'll first start as if it were flat space. So if we were to have a bunch of data points of a shape, where every single point is a shape, and that space were flat, we would use a common analysis for linear space, which is linear regression. And you might have heard about this in science class. It's essentially where you learn the line that best fits these data points. And when you learn the line that fits these data points the best, you're essentially learning the slope and the y-intercept that will give you the line that will go through these points. But for a curved space, we can't do linear regression, because linear regression, it would give us something completely meaningless on a curved space. So instead, we have to do geodesic regression. Geodesics are essentially the same thing as a line on a curved space. So my research develops geodesic regression techniques so that we can predict future shapes, which essentially means potentially predicting disease, and so that we can quantify how much a shape bends or stretches over time. And this second one is a particularly new technique that Nina and I have developed. So, what types of questions does this even answer? If we can quantify how much a shape bends or stretches over time, like, who cares? That's a great question, by the way. Who cares? It would answer questions like, is this hippocampus responding normally to progesterone exposure? Like, by studying a bunch of brains and a bunch of hippocampi and measuring how much they bend and stretch over time, we would be able to know what the normal amount of bending and stretching is. And then we would be able to associate abnormal bending and stretching to particular diseases. So then, by measuring the bending and stretching of a hippocampus over time, or in response to progesterone, we would then start to be able to answer questions like, is this hippocampus showing early signs of Alzheimer's? Or if this hippocampus is in a pregnant woman, is this hippocampus showing early signs of postpartum depression? Or, the list is endless. Nina and I have already developed this method on 2D cell shapes, and we published this in a workshop at a machine learning conference last fall. And the next step is to just generalize it to brain surfaces. So a lot of the work has already been done. Not to jinx it, but yeah, so with this work, we're hoping that we can start to help the Women's Brain Health Initiative answer some of these basic questions about menstrual cycles, pregnancy, and menopause. So to conclude, women's brains are under-examined due to historic bias in brain studies. This has led to women being underserved in healthcare, and the Women's Brain Health Initiative is trying to change this trend by measuring a lot of human female brains. And the BioShape lab is aiding this effort by contributing cutting-edge shape analysis research, which will help model how structural changes in the female brain correlate with sex hormone fluctuations. Before I conclude, I'd like to acknowledge some of the people who have made this research possible. The Ann S. Bowers Women's Brain Health Initiative is made possible by a generous donation from Ann S. Bowers, and my research in the BioShape lab is supported by fellowships from the National Science Foundation. If you want to help, you don't have to donate money. You can just sign up to get your brain scanned, or encourage others to get their brains scanned. We're specifically looking for people who are naturally cycling, so maybe no birth control, planning to become pregnant, or approaching menopause. Thank you everyone again for coming, and thank you Brass Bear Brewing. I will take any questions if you have them.
Have you had your brain